It's my pleasure to introduce Zunlong. So he's a postdoc with John Briggs at the Max Planck Institute for Biochemistry. And so he's going to talk to us about Cryo-ET and sub-tomogram averaging of envelope viruses. Thanks, Zunlong.
Thank you. I'm also a graduate student with Liz when I was doing my PhD. So, right. So, well, welcome everyone, and thanks Liz for the invitation. Much appreciated. So I'm going to talk about sub-tomogram averaging a bit, and I'll show a specific examples here we go. So I'll start with a very brief introduction about Cryo-ET, apophyretin structures, and then follow up with two stories on the SARS-CoV-2 variant spike and the distributions and conformations. I will briefly, this is not working so well, briefly introduce the work I've done during my PhD at Liz's lab, and I finish with maybe in situ single particle cryo-EM on those native variants. So cryo-EM that we all know has evolved so much. This image here shows like the atomic structure of apophyretin, 1.2 angstroms. I think it is truly amazing. However, we do not have the context we wish to achieve, because any structures of proteins, they will function inside cells that will be lost if we do it this way. And however, there is a solution to that. It's called cryo-electron tomography, or Cryo-ET. That's this workshop is about. Is that I'll show you an example of the evolution of Cryo-ET instead of evolution of cryo-EM. In 2007, Liz, this is work from from Liz when she was doing her postdoc with Grant Jensen's lab, that you can see this capsid is the capsid protein of HIV that's been solved to 30 angstroms at that time. Now, about a decade later, the Briggs lab, or Briggs, that solved the same protein to sub-nanometer. That really allow you to see these secondary structures. And then recently, that's been solved to like 3 angstroms of the same protein complex. So you can see this evolution of Cryo-ET that has also going much faster than we would think. So Cryo-ET not only just be able to solve the structures, because that's part of it. The other part of it is that the beauty of it is that they can resolve the structures in situ. What that means is that if you resolve it in situ, you must have the context of it. That context is you can put things back. When I say I put things back, is that you can put each hexamers or pentamers to where they are from, orientations, positions, and how they interact with each other. So that is, my point of view, is the beauty of Cryo-ET, that you can simply not get from other methods. So that we can obtain the context of protein spatial organization, the lattice. So this is a movie about how that works in a nutshell, I guess. So this is an example of herpes virus. You have this frozen viruses and at cryogenic temperatures. And then what you do is this is frozen on the grid, and you load that grid onto the electron microscope. And then this is the grid. What you see is that you can see different tilt of images collecting from -60 to +60, in this case, about every 2 or 3 degrees. So you get essentially a series of projections of the same sample. And you can combine them all to reconstruct what we call a tomogram. So those individual images will be called tilt series. And after reconstruction, you get a volume that's a tomogram. So this method, using back-weighted projection or other ways to do it, that you can get a like the 3D volume of this herpes virus. And then you can see after segmentation and the process, and you can see individual slice of this capsid. Oh, this one is the membrane protein, and the other one inside is the capsid inside. So now the story of my work. about SARS-CoV-2 spike. At the beginning of the pandemic, the SARS-CoV-2 spike has been determined by single particle cryo-EM by various groups that you can get, like 3 or so angstrom structures from the purified individual isolated proteins. That you can see on the right side is a tomogram of these isolated proteins that I collected at the earlier stage of the pandemic. What you can see is that, again, you do not have the context of how the spikes function on the whole virion, because that's supposed to be where they should function, how they infect cells. And you don't know how the spikes distribute relative to each other one to next. You don't have how they organized relative to the membrane. Like, you don't have any of that. You just get the structures, which is very important for sure for development of vaccines and drugs. About 15 or so, I guess, a decade and a half ago, people have used, like the whole variants to study coronaviruses, not the SARS-CoV-2 because it's not there yet. But they use, like the SARS-CoV-1. What you see here is that, a variant with spikes protruding out, and even at the time they have done reconstructions of the spike that you can see. It's essentially a membrane with a volume projecting away from the volume that essentially does not have any more details than this that prevents us from a better understanding of the molecular interfaces. So when the pandemic started, we were very fortunate to get the samples of the SARS-CoV-2 from our previous collaborators at University of Heidelberg. And so we started to get a sample and do tomography. The goal is to achieve whether we can get the spike structure from the virions instead of from the isolated proteins. And you can see this left is the 2D tier series. On the right is a tomogram. You can see each tomographic slices, you can see the spikes really well protruding out from the membrane. If you notice more, I guess we can go over a bit later. That's different properties of the spike. So yeah, that's the collaborator in the biosafety level 3 lab in Heidelberg. And we were shipping the tools and grid boxes and stuff to them at the time so that they can prepare samples and stuff like that and ship it back to Cambridge, UK. So here, I hope this plays. Here we go. Is how we zoom in to collect data, and this is like the hole. So the hole is about 2 microns. You keep zooming, and you can see the spike of the virion actually, and you can see the gold fiducials, and you start to collect. You find the region you like, and you start to collect the tail series, right? And after you collect tail series, you start to build the tomograms, and you can see the slice of that viruses at different how we collect it. So just a very side story to share. This was actually me when I was collecting data during the pandemic. That was April, probably. So you're not allowed to go into the building because it's pandemic, but people work on coronaviruses, they can go into, but because just maybe 1 or 2 people in the whole building, you need to wear this oxygen sensors just in case anything happens, and people will be notified and I guess rescue you. So that data set is very, very, very special to me in a way that I spent 6 days collecting it. And it's very diluted. The sample is from the virus supernatant, which I'll go next slide. You have to find them, and then find them by collecting 136 square maps. That essentially is the whole grid. And I only found like 200 something tomograms. So those are the different samples we experienced. So the first sample is obviously you can infect the cells, and you can culture cells on the grid, and then you can plunge freeze it after a certain time point of infection. And from here you can see this protrusion of viruses, a protrusion of cell, and you can get like those viruses out. So this we call it this virus infected cells. And the second sample here is the supernatant sample. What that means is that you take the first sample, virus infected cells, you take the supernatant only, and you can imagine it's just too much volume, right? Because it's very diluted. You don't you don't centrifuge it after that. What you do, you get rid of the cell debris, like a low-speed centrifugation, and then you have essentially like a very diluted supernatant, and you use that to do imaging. So you put, like 4 microliters onto the grid. You're essentially looking needle in a haystack, but that was the hope that we might be able to find it. That was what we had at the time. And the third sample is you take the supernatant, you centrifuge it 1,000 times, and sucrose cushion, you get the virus at the bottom of the pallet, you resuspend it, then you get a lot more. You get like 1,000 times, you have no problem finding viruses. And the fact is you have 2 more, way too many. So those are the 3 samples we have done, and then we decided to move on with the second and the third sample because the first sample is really hard to find those regions and to get a large data set. So I will talk about this first and the second sample. The first sample we did most of our tomography analysis on. So we collect data, we build the tomograms, and that's the tomograms, and then we start to think about, okay, how we gonna get the structure out. So the first thing you do is you need to pick the particles however you do it, right? At the very beginning, you have to manually pick it so they get a template, because there are many structures out there, but we would like to get the structure with the membrane on from our data. So you pick each individual particle, like here, 3 particles here, and then as shown on the pipelines on the top here. So you pick your particles, and then you are going to align them one against the reference, and you align them up, and you average it, and you get a higher signal-to-noise ratio averages out, right? So after you get this, so initially I picked about 60 or something, spikes from, I don't know, 2-3 tomograms, and then you pick 2 points so that you pick the top and the bottom, so you have a orientation already. So you average them up, you get an initial reference. It's not lovely, but it will do the work to use it at this to serve the reference at this stage. After you have this one, remember we have many more tomograms, right? It's a lot of work to pick each one of them. As Alberto said, picking is very tricky and it needs to be robust. So what we did was we defined the center of the virus, and then we defined the radius of it using this camera. And then after we define the center, define the radius, we can define the sampling spacing, and we can just get loads of it around the sphere. By doing that, we can get locations and orientations, projecting away from the membrane. So you get, this is a bit oversampled, obviously, for this spike, but you get the idea is that, you sample a lot more so that eventually they will align to this, true spike positions. So you get, so after that, you do iterative refinement with all the positions you get from the larger data set using the reference we obtained at the very beginning. And then you can get iterative refinements. This is the iterative refinement of the spike at different iterations. You can see the structure evolves. So we got about 7 axon structure, at least at the central helices regions. And the next question we ask is, okay, so what else can we obtain from after this? So we started to think about the receptor binding domain that is this RBD, or we call it the receptor binding domain, that is on the top of the spike. That's the region binds to the host cell to start the initiation of the membrane fusion process. It has a very important function, but it's also known from previous studies that this region, this highlighted in magenta, has different conformations, receptor bindings down or up. So those receptor binding domains is known to be flexible. So we observed like three different conformational states with like 0 receptor binding domains open, 1 up, and 2 up. And we also have 3 ups, but very small portion. Sorry. And then also we have observed this very long, another conformation is called post-fusion. The post-fusion is essentially the pre-fusion but cleave the top and then extend it away. That's after membrane fusion. So very, I just want to go over this step really quick here, is how we do this classification of this region of this receptor binding domain. So we up to this point, we got 7.7 exon structures that we assumed it is a threefold symmetrical protein, right? But we also know that each region can be different. So how do we, we assume C3, now we have to untangle this C3 back to, where it should be. What we did was essentially, you can rotate this aligned positions to, 120 degrees and to 40 degrees. You essentially symmetry expand it. And you can get, you can imagine if a spike has no receptor binding domain, doesn't matter how much you rotate it, 120 or 240, it will be the same, right? But if you have one that's open, it will be at different locations. And when you have two open, that's also will be at different locations. So, and the three, again, is going to be the same. So the idea is that you will be focusing on only one position, let's say here, right? At each time you just focus on this one, and this one here as well, and here as well. This will be absent, this will be present, right? So this one is present, absent, absent, present, present. So like you just do it this way, so you figure out, which particle has 1 or 2 or 3 or 0 opening position. But then you go back to track where they're from, and you can understand how many monomers of the spike that's in open or closed conformation. And you can go back again to the trimer, to the spike, to figure out what's the percentage of each one. Now, we have done the structures, we have done the conformations. The next question is we observed that the spike is very tilted towards the membrane. So imagine this is the membrane of the virus, and normally a spike will go kind of perpendicular to this membrane, right? However, like for influenza virus like HA or like Ebola or other virus, they just go this way, roughly, maybe a little bit tilt. But for coronavirus, we observe super tilted, can even go up to really high tilt angles. So we ask, okay, can we characterize this tilting angle on the viral membrane? And the way we did it was that because we already know the center of the virion, we also know how big the virion is, like the radius of the virus. We also have obtained information about how this particular spike is oriented in 3D space from subtotal averaging. So we can calculate, you can use these two vectors. One is from the center of the virus to where it's closest point, and then the other vector is this orientation of the spike itself. So you can calculate the angle of it, and by doing that, you can calculate each of the spikes, and you can get the angles of how much tilted the spike is relative to the membrane. And here are examples of 14 degrees, 32, 45, and 76 degrees. You can see it is truly super tilted relative to the membrane. So now, if you pay attention to what we had before, which is that, for example, this spike structure is that you see this spike structure, we don't have anything below that, like the membrane is lost, everything's lost. It's because this region here is very it's very flexible. After that point, it's very flexible. So we group this different tilt of angles of spike, we average them, and we can get spike at different tilt angles as well as different, so that it can allow us to understand at which point it starts to tilt. So that help us to understand like how the viruses, when they are tilted, they will be able to, I guess, facilitate the process of receptor binding domain so that they can have easier access if the receptor binding domain is there. And now we have obtained the structure, we have obtained the conformations of the structure, we have analyzed the tilt angles of the spike relative to the membrane, right? Now we can build everything together, we'll put them back to where they are. This is truly the power of Cryo-ET and subtotal averaging, is that you can understand each one of the spike relative to each other and how they are relative to membrane and or relative to the next one. And you found that the spikes are actually very sparsely distributed on the viral membrane, and about 1 trimer per 1,000 nanometer squares per tremor. This number is actually really low. Influenza virus, for example, has about 10 tremors per 1,000 nanometer squared. So it's much more abundant in this case. And you can also see there's not much relationship between each spike and their confirmation, how they tilted. It's not much relationship between that. Okay, so I have shared with you this SARS-CoV-2 spike story. Now I just want to share with you a very small story that, well, not a small story, but like, what I've done during my PhD with Liz, of the respiratory syncytial virus. This virus has two, well, not two, but like it has two different morphology. One's filamentous, it's really long actually. One is spherical at the time. It was kind of, so we want to know, I guess one of the questions we were trying to ask is that, what is this morphology of this protein, and also what are the conformations of the fusion glycoprotein on the surface, right? We do cell culture on the grid, and then we do infection, and you can see this this is a cell here. And we also do like correlative microscopy at the time to figure out which cell is infected, which cell is not infected. You can see the viral filaments coming out of the plasma membrane. So, and then we do data collection. So this slide is essentially to show you like how we do data collection on a like a very complicated sample instead of like epiferritin, I guess. So what our target is, we want to understand where virus assembly happens, right? So we need to target them, find them, and collect data there. Anything else, I'm not interested. So you can see this is like a big cell, A549 cells. This is the outside the cell, and this is the border of the cell in this yellow color. You can see here in this region, as zooming in here, that you can see this is the viral filaments coming out, and then you can start to pinpoint to that location. So this is all montages. This is our 2D images, just montages, big montages. And you can start to collect data at these regions, and then you can start to analyze viral proteins and then host cellular proteins in this case. Yeah, I was going to talk this slide maybe later, but we can talk about this one briefly. So the target of the data collection is very important in a way that if you have, as someone made a point, that if you don't have a good tomogram, none of them, after that will happen. So what this means is that if, imagine you have collect data, like here, your focus will be, your focus will be like somewhere here, for example. And then in this case, I had another example to show you, right? Is this a good location to collect my data if I wish to collect, viral filaments? I'm asking, why not? It's too dense. In what way? They're on top of each other, right? So there are, you can see like projections of viral filaments on top of each other, indicating that essentially the ice here is thick, multi-layers of proteins or viral filaments on top of each other is just not a good idea to collect there. Overall here, okay, imagine your magnification goes up, so your field of view is smaller, right? So now we're here now. Is this a good idea? Probably okay, right? Then here, if we go even bigger, this is also a good idea, I think. So essentially, you need to be very selective when you collect tomograms if you're not going, to collect thousands of tomograms. But in cases like this, like viral filaments, like, stuff like, especially for like assembly sites that I had earlier, those are rare events. When you have it in a good location, you probably want it, but you want it, it's not like selective still, always selective when you collect tomograms. You also, say example, this one, right? For example, if it's good, right? Okay, everything's good, gold is good, everything's fine. But there's a big chunk of ice here. When I tilt axis this way, I probably wouldn't collect it there either, even though it's so good, but I'm just gonna leave it because I know I will not get a good tomogram afterwards. So So here is an example of the virus assembly that we found at the plasma membrane at the edge of the cell. I'm going to show you a segmentation of this lovely movie. I spent one of my Christmas 7 days on it. So you can see this different, virus and cellular proteins on them, like microtubule and actin, and these are viral proteins. Yeah. So, and also you can see this is not even in the hole, it's on the carbon, right? But in cases like this, cellular tomography, when you have a really lovely target, you just take it, but also be selective still. Okay. So that's about that's the end of it. Well, it's not the end of it. It's the I will show you two more examples actually about another two projects I took. So this is the Ebola virus filamentous particles. You can see this super filamentous, and you can see this glycoproteins coming out of it. What we did, or I did on this study is that I want to analyze the fat fragments bound to this glycoprotein. And obviously, we pick the particles on this and the substructure of it. And again, another example to show the power of Cryo-ET is that you can put them back to where they are from, and then you can, kind of understand. In this case, is we won't understand if the fabs will be kind of sterically hindering each other if you have three fabs bound to it. Like if space-wise, we'll have enough space to accommodate three fabs on a single glycoprotein, and seems like it can, but not sure about it. So the other one is the HIV project that I did. So this one is matrix protein under the membrane, right? So this one is I think by far one of the most challenging subatomic averaging projects I've done. That matrix protein is very small, but obviously we don't have time to go through details and talk about it. But eventually it got solved to sub-nanometer resolution, and you can see the lattice spacing. I was going to maybe show a bit on Thursday sessions if we got time. Okay, so the last couple minutes, I will just talk about this part is that a lot of people have started to do tomography, right? Cryo-EM people. I was thinking in this particular project, or the particular sample, Alberta said that we make most use of our sample we got, right? Using any method we can. So this is the project I did on the SARS-CoV-2 spike that you can see. I just show you data collection, parameters we use. So this is one image that we use this parameters. On this image, if you zoom in, just this image, and just zoom in, and you can get this kind of, details about it, so you can eventually know which host has these viruses in it. It's very important because if you take less images, then you can get your speed higher. So you just need to tune your parameters correctly. So this sample we had was the concentrated sample that I talked about. So we have supernatant, we have a concentrated sample, and we can do essentially a single particle approach to collect the data. And then you can see the spikes on the protrusions of outside the membrane. And we did data picking the particles, and then we did class 3D straight away. And usually you can find a good class that has the spike structures. And you do refinement. This is truly single particle, just single particle, and you can get high resolution structure of the spike on the virion as it is. So I just want to point out that with the sample you have, you can try different ways to do it and get high resolution. But what we don't have using this way is we will never have the information we have provided, like the tilt angle, those kind of stuff. So I think it will be a hybrid approach will be great. Okay. To summarize, I will share the Cryo-ET evolution, and I've shared stories on the SARS-CoV-2 spike and how they distribute, and also this assembly of this RSV. yeah. So with that, I'll I'll acknowledge people from the Rat Lab. There's a lot of people doing my PhD that has helped along the way. It's really good. There's a list. And also people from my postdoc lab at the John Briggs lab, and all our collaborators from different providing resources as well as collecting data. Yeah, this is the Briggs lab. We play a lot of foosball. Oh, it stopped somehow. Yeah, I don't know, but okay. I'll take questions from here. Thank you.
Yeah, I had a question. How you dealt, or two questions. One question is how you dealt with maybe oversampling the particles in the early step of the SARS-CoV spike protein on the virion. Because I think in like single particle, we have this cool like 2D classification to clean out bad particles or junk at the beginning. Curious how you dealt with that.
Right. So obviously, this is way more than I should sample. But let's say, assuming I only have 2 spikes on the surface and I sample, let's say 200, right? How am I going to clean that afterwards? Essentially, I treat each position as a Sub-Tomogram, and I align to the reference, right? Allow it to shift as much as it needs to go to the true spike. What happens after iterative refine or after alignment, right? Is that there's a spike here. The one next to it will essentially go to here if your alignment parameters about everything is right. They will go here. So you will get a cluster of, let's say, 5 points at that single position, clustered super tightly. Occasionally, you do get the clusters of points here that the spikes those ones just cannot find that one or that one because they're just too far away. So you get points here, but scattered. They won't be clustered. So the way to do it is the first step is to get rid of the one that's not clustered because they won't be spikes. And after that, you just keep one of the ones that's clustered. I have also, set different criteria to do the clustering. But yeah.
So it doesn't like remove your reference. Like if you align to the reference with all these particles that aren't spike proteins, you still, I guess your classification was strong enough that you still had some classes with the spike protein.
It's not classification. It's alignment.
Okay.
So essentially you align this particle to that one, right? But because there's no spikes here next to it, it will not be aligned.
Okay, I see. It's not like a refinement strategy. It's an alignment strategy.
It's alignment strategy.
And the other just quick question was, you had a movie with the spike protein kind of rotating. Was that just a morphing between different volumes?
That's right.
Thanks.
Yeah, it's just morphing of different volumes. That's right.
Okay, question for you. On your collection parameters slide, what's your feeling about objective apertures?
Apertures. Apertures. I think it's good. I have it in it. 70 microns or something. Yeah, I have it. I think it helps. I mean, I'm not going to get 2 angstroms, so. Is that?
Hi, my question is regarding the point that you made about the receptor tilting on the SARS-CoV-2 spike on it favoring receptor binding. I was just curious on how that is widely known, of whether there is structural information to suggest that as well, or?
So how does this structure, results helps us to, I guess it might help, the binding process. That I mean, it's a really good question. It's a guess. But you can imagine, right? Because the spike can tilt, right? If this receptor is here on the cell, that probably won't move, while the virus is moving around on the surface of the cell. If it can tilt, it will just. But that's a guess.
Okay. I guess I was just curious because earlier you were talking about other examples of viruses that didn't have that tilt. I think you mentioned Ebola as being one of them. So
right. The other viruses, for example, so this one, right? Let's see. Actually, so there are two things happening here. One is that the spacing of the spike are further apart from, like, let's say influenza virus 1 and 2. The spikes on here tilt more than influenza virus, right? So those are two things kind of go, I guess, in a way together, kind of help us understand, in my opinion, it's called a avidity effect, I guess, in virology, is that when one binds, the other will bind easier or better. It's like the sequential effect. So in this case, we don't have a avidity effect because we're too far away from each other. A avidity effect needs you to be really close to each other so that you can, affect the next one. But we don't have it. But what we also know is that the spike to the receptor binding, the binding affinity is much higher than flu. So it kind of goes together, those three things, I guess.
okay, thank you.
Hi, really, really fantastic talk. So you mentioned in the beginning when you were talking about your process for getting the sample that you that it was hard to get images with the infected cells versus like centrifuging them. So I guess I'm just curious if you could go through doing this data collection on infected cell, like how you can circumvent the issues aside from just centrifuging and pelleting.
Like here,
yes.
If you want to go this path, so my PhD work with Liz was essentially this, is that we collect, virus assembly at the edge of the cell, and that's the only thing we're, I guess, at the time interested to understand these different assembly stages, right? What you do, the best you can do is you really try hard on the optimization of your sample. That means you do a time course experiment of the infection, you get the optimal, maximum, assembly events as your sample you have. Like go back to your, biochemistry or virology or cell culture, go back to that to burst the chance of this. Then you have a better chance to look for what you want. Other than that, I don't have it. Yeah, I mean, claim, but that's a lot of work as well. You just try harder in your sample. That's, yeah.
Hi, very nice talk, Zunlong. I'm just curious, is the flexibility of the spike protein has any connection with the receptor binding domain open or in closed conformation? Like, is it the tilting angle related with this?
Yeah, so this, I mean, that's a brilliant hypothesis for sure. It seems not. So we have, we have this information of how much the spike tilts, right? But we also have information about that same spike, whether open or closed, right? They're not so much correlated. Yeah, it's a good hypothesis.
All right, Zong, I was going to ask one more technical one, and then I think another that's a biological question.
Okay.
The first one was when you use, I'll scroll up a little bit. Which reference did you use to line the sub-tomograms and the 3D reconstruction? Did you prepare an initial model for the PIC tomograms? I think this refers to the spike section.
This one, right?
Yeah.
So what I did was I pick 62 or something, 66 sub-tomograms by 2 points on the head and on the tail, like a 2 points. So I define this 60, spikes and orientate them essentially in the same way like this, and then just average, no alignments. Because, yeah, in order to do alignment, you need a reference, right? But if you don't do alignment, you just average, then it's fine. So you just average them all. So you get a blob with a membrane and a blob on this way. I use that as my initial reference. And then from there, you can use that blob to do alignment of this 60. And then you get that blob. And from there, you keep going.
Sounds great. So it's all starting from the data and not.
It's everything is from, it is, yes.
And then the biological question was, does HIV capsid migrate through the nuclear pore complex? This has been shown at Max Planck, I believe.
Yeah, well, it's Max Planck. Does the capsid migrate to the nuclear pore complex? Well, it's not really relevant to what I do, but okay, so there's a study that they show the capsid, right? The mature capsid goes into the portal of the nuclear pore complex. They kind of like in between. That's actually a study with Hans Georg and Martin Berg at EMBL. Like they show that. I mean, it's not anything I do. I actually work on that project. Failed, but yeah. I mean, it seems like they can.
I think the continuation of it was reassembling after the reverse transcriptase, then taking the integrase to the nucleus in the SID capsid. Can you explain how the cyclophilin A is able to facilitate this deassembly and reassembly? I think it may all be far upstream from.
Do I talk about it or okay? Okay. I mean, that study actually I can answer it, but it seems. Okay.
Any other questions? Okay, well, let's thank Zunlong again.